This is the start screen for the Athena image processing software that accompanies your vScan Hermes high-content imaging microscope. In this video, I will describe how to load data acquired on the Hermes. Similar to the Hermes software, we select the user account from the list displayed on the left. Once the user account has been selected, we're taken to this, the data loader screen. By following the tabs along the top of the software, we can see how the software guides us through the data analysis workflow, beginning here with the loader screen selected in light gray. Next, we will optionally be selecting an analysis protocol. Then we will move to the analyzer screen, where we can choose the analysis application we'd like to use. Finally, we will be taken to the results screen, where we can explore the data we have extracted. Alternatively, we can begin on the Results tab to begin visualizing the images and optionally export montage movies or other image processing steps. Back here to the loader screen, we are provided with two options for data selection. Either we can select data that is listed under our user account as shown here, or we can browse to another location on the computer or on the network to select a data folder of interest. As you can see, I currently have 3 datasets present in my folder. The dataset on top has a green dot next to it, indicating that it has not been previously analyzed. The ones below it have a purple dot indicating they have been analyzed. Double-clicking on the name of this dataset, we're taken inside to see the original raw data shown with the green dot, and the previously performed analysis shown with the purple dot. On the right, we can see a summary of the imaging parameters that were set both during image acquisition and the analysis parameters that were used during the previous analysis. To select this previously run analysis to perform again, I will simply select Load Experiment. Here, we are now on the protocol screen. I'm able to select a protocol file which contains the image analysis parameters that have been previously defined in a previous imaging session. The protocols present in the shared folder are common to all users of the Athena software. Alternatively, I can select the user folder to identify protocols unique to my user profile, as I have selected a previously performed analysis. I can select Use Current Protocol or Define New. Now we are present on the Analyzer screen, where we can see the list of potential image analysis applications present on the left. I can scroll down to see the complete list and choose between different applications if I so desire. For instance, I can choose cell count, cell morphology, nuclear translocation, and in each case, we see the buttons representing color channels that need to be assigned to the different objects identified or signal measured in the selected application. Here I'm going to select protein expression, and we see there are 3 structures that we need to identify. The first being the protein. This is the channel that we will use for integration of the fluorescence signal intensity to identify changes in protein expression in our experiment. We have the nucleus structure, which is going to be defined in blue, and this is obviously to identify the nucleus of the cell. The cell cytoplasm is then identified in green, and together these form the objects and parameters needed to begin the analysis of this dataset. The last screen that we're going to go through in this video is the Analysis Setup menu. Within this menu, we can choose the wells within our multi-well plate that we want to include in the analysis run. We can do this by simply selecting a subset of the wells shown in cyan and click the Set Analysis Region button. The wells shown in cyan will be included in the analysis, while the wells shown in green will be excluded. I will select all of them again Optionally, we can set positive control wells and negative control wells with these two buttons respectively. Here, the positive and negative control wells have been previously defined using the Hermes software. Optionally, we can load tags, which are comma-separated value files that define the concentration and treatments performed for each well in a multi-well plate experiment. Creation of tags will be defined in a different video. Lastly, we have the dataset info, which we can optionally define here, where we can initialize the name of the analysis folder and write notes to keep in mind when looking at the results. So we click on the Done button to save all of the settings we've uploaded and are brought back to the Analyzer screen. This is the end of this video, and next we'll be taking a look at the image analysis steps themselves.